www.steep or on the Town of Barnstable's Channel 18 video on demand platform. My name is Cheryl Osimo. I'm the Executive Director of the Massachusetts Breast Cancer Coalition, one of STEEP's community engagement core partners, along with Sierra Club Cape Cod. I am also Cape Cod Outreach Coordinator for Silent Spring Institute. Launched in 2017, STEEP is a research program funded by the National Institute of Health Sciences. The program is led by Dr. Raina Lohman and the University of Rhode Island in collaboration with the Harvard T.H. Chan School of Public Health and Silent Spring Institute. On behalf of the STEEP team, I'd like to welcome you here today for an informative discussion on the health effects of PFAS in drinking water. Our speakers today are going to talk about the effects of PFAS on the immune system, childhood vaccinations, and susceptibility to COVID-19. A special thank you to STEEP for organizing this series and to the Town of Barnstable, and most especially Mark Ells, our town manager, here on Cape Cod for their continued partnership. And now I have the pleasure of introducing Mark Ells, town manager for the Town of Barnstable. Mark has worked for the Town of Barnstable for 31 years and is currently the town manager. He has also been an adjunct faculty member at Cape Cod Community College for 19 years in their business administration program. Mark serves on several regional and statewide committees ranging from environmental, human services, education, and transportation issues. Mark holds a bachelor degree in engineering from Norwich University and an MBA and an MPA from Western New England University. We're so pleased to have Mark join us today, and now I'll invite him to please say a few words. Mark? Mark?
I can't hear you, Mark.
Mark? Okay.
Oh.
It's okay, Mark.
You would think at this point in the use of Zoom that I would know to unmute. So I apologize.
Don't apologize.
And, and good afternoon, everyone, and welcome. The challenges of technology. I want to thank you for joining us for a virtual conversation of PFAS and its presence in U.S. drinking water, including waters here on Cape Cod. On behalf of the Town of Barnstable, we welcome you to this virtual monthly webinar series, Let's Talk PFAS. It's no coincidence that Barnstable has partnered with Silent Spring Institute for decades. Our shared core values that everyone should live in an environment that's safe and free from pollutants of concern is at the foundation of our relationship. For those of you who are familiar with Barnstable, it's our mission to maintain a high quality of life for our residents and visitors, and our water resources are a critical part of that quality of life. So we are very pleased to host this and any event with our partners. And at this point, I will turn it back over to Cheryl Osimo. Thank you.
Thank you so much, Mark. We're appreciative of Mark's support of the STEEP Superfund Research Program and for the longtime support of the Town of Barnstable, which has paved the way for us to be able to be doing this work. And they have been so supportive of all the research we have been conducting here on Cape Cod for so many years. Now I'd like to introduce our speakers this afternoon, Dr. Carmen Masserlian and Dr. Philippe Grandjean. Dr. Carmen Masserlian is an assistant professor of environmental, reproductive, perinatal, and pediatric epidemiology at the Harvard T.H. Chan School of Public Health. Her research is focused on examining the extent to which environmental exposures affect a couple's ability to achieve conception, maintain pregnancy, and deliver healthy offspring. Dr. Masserlian is also actively collaborating with leading bench scientists to conduct translational research to examine the underlying biological pathways that may lead to infertility and adverse pregnancy and child health outcomes. Her ultimate goal is to use this evidence to inform clinical practice and improve health for mothers, fathers, and their children. We're so honored that you're with us today. Thank you so much. Dr. Philippe Grandjean is an adjunct professor of environmental health at the Harvard T.H. Chan School of Public Health and serves as professor and chair of environmental medicine at the University of Southern Denmark. Dr. Grosjean conducts international research to assess adverse health effects that are linked to exposures to environmental chemicals, especially those that occur prenatally during highly vulnerable early life stages. He serves as the STEEP Center co-principal investigator and principal investigator of Project 2, which is carried out in the Faroe Islands and is aimed at exploring the adverse effects of the immune system in children. Since 2013, he has been a member of the European Environment Agency's Scientific Committee, and he also serves on the World Health Organization's European Advisory Committee on Health Research. We'll begin with presentations from both speakers and have reserved time at the end for questions from the audience. We encourage you to use the Q&A feature in Zoom and submit your questions throughout the webinar. We may not be able to get to all of your questions, but we'll take all questions into consideration and we'll do our best to answer them in future webinars. Thank you, and I'll now turn it over to Dr. Carmen Masserlian.
Thank you very much, Cheryl. Thank you very much, Mark. It's my pleasure to be here today. I'm going to wait for the presentation to load. Thank you, Sarah, for being in charge of the switching of slides, which always adds a little complication. So thank you very much again for welcoming me here today to this wonderful webinar. I'm very excited to be here. Well, the title of my presentation is about perfluorinated chemicals, or PFAS substances, as we know. And I'm going to be focusing a little bit on the immune function in children. I'll present some new data. I will also talk to you a little bit about what these chemicals are and how we're exposed and why we need to be concerned about them with respect to children's health and to pregnancy. So with that, we can advance to the next slide. I have no conflicts of interest to declare. Thank you very much. We could switch to the next slide. So to the background. So the background of this presentation, Cheryl, we could focus on the next slide. Thank you. So just a little brief background. So per and polyfluoroalkyl substances are environmental toxicants of urgent governmental and public concern over their near universal human exposure and adverse health effects. Despite decades of research, the consequences of PFAS exposure for human health have remained strikingly unclear. And PFAS chemicals are really a family of chemicals of high production volume, synthetic and water and stain resistant chemicals that are used in countless consumer products, including nonstick cookware, textiles, and food packaging. And PFAS can be found in the water we drink, the food we eat, the air we breathe, and in our homes, schools, and workplaces. The chemical properties that make PFAS useful in so many applications also cause persistence in the environment and accumulation in the body. And we know that the half-lives for these chemicals range from between 2 and 8 years. Indeed, PFAS have been labeled what we call forever chemicals. And the general population exposure is ubiquitous to chemicals such as perfluorooctanoic acid, or PFOA, or perfluorooctane sulfonate, or PFAS, which are the two more common ones that we've studied. And these are detectable in the serum of more than 99% of the United States population. And this is not just in the American population, but throughout the globe. PFAS have also been measured in human amniotic fluid and in cord and neonatal blood. And although the two most widely studied PFAS and PFOA were voluntarily phased out by manufacturers in the early 2000s, human exposure remains prevalent due to widespread environmental contamination, such as that we have seen in Cape Cod. And there is concern regarding the dozens of newer PFAS that have been used as substitutes to PFOA and PFAS, for which research is lacking. So we can go to the next slide. Thank you. So exposure pathways for PFAS include dietary ingestion, house dust ingestion, indoor air inhalation, and dermal absorption. And ingesting contaminated food and drinks is the dominant pathway of PFAS in the general population. The major sources of PFAS in human blood are seafood consumption for PFOA and PFAS and PFNA, and consumer products such as carpet and nonstick pans and cookware for PFHXS. Drinking water may contribute up to 20% of exposure levels, although it is highly dependent on the geographical location. And although direct exposure of PFAS from consumer products can be reduced through shifts in production, exposure driven by contamination in groundwater from, for example, firefighting foams and accumulation in food chain can persist over a long period of time. Similarly, we know that transplacental and breastfeeding passage of accumulated PFAS concentrations in mothers is really inevitable. Therefore, we need strict national and international regulations to stop the vicious cycle of exposure to these persistent chemicals. So we can go to the next slide. Thank you. So where do we find these chemicals? So as I mentioned, there are numerous different exposure groups, but PFAS can be found in everyday consumer products such as carpets and upholstery, waterproof apparel, waxes such as those that you found on your hardwood floors or your skis, non-stick cookware, grease-proof food packaging, paper packaging that you get takeout in, for example, dental floss, cosmetics, and paints. We can advance to the next slide. Thank you. What really concerns us, however, is that it has been demonstrated that children have a higher body burden of exposure, and they have been found to have elevated serum concentrations of PFAS compared to adults. And this is thought to be due to mouthing behaviors, to time spent on the floor closer to dust sources, also as a result of their different body size to surface area and the intake ratios across ages. And exposure, as I mentioned, also occurs through placental transfer or through breastfeeding. And neonates and breastfed toddlers are probably the population group exposed to the highest concentrations of these chemicals, and they are a vulnerable population that we need to be concerned about. So into the next slide, thank you. So in addition to this, what we do know is that early childhood is a critical window for many important health endpoints, including the immune system. And the immune system gradually matures during infancy. So early protection is provided by the mother through passive antibody transference through the pregnancy, through the placenta, and through the milk in the postnatal period after the baby is born, if the mother chooses to breastfeed. Then young children are directly exposed to infections over the early infancy period, and this allows the infant to develop an innate and adaptive immune system that helps their immune system mature. The accumulation of this immunological or immune memory is an evolving feature of the adaptive immune response. And as a result, young adults suffer fewer infections. This memory persists into old age, but then it may fade over time, in which case older adults tend to be more at risk, again, to infections. And alterations and deviations during this training period that occurs in infancy and childhood may actually have long-term consequences that predisposes this child to future infections, to autoimmune diseases, or even an increased risk of things like cancer. Okay, the next slide, thank you. So what do we know about the mother and infant, or the mother, fetus, infant is highly interdependent. As I mentioned, the fetus receives antibodies from the mother through the placenta, and then the neonate also receives transference of these antibodies through breast milk. And as I mentioned, the adaptive immune system is very antigen specific, and the variation in this response can change as a result of exposure to chemicals such as PFAS. And the acquisition of innate and adaptive immune system function in infancy may be influenced by PFAS exposure both in pregnancy as well as early infancy. Could switch to the next slide. So now I'll present to you one more slide, some recent evidence. Actually, I'm sorry, I'm going to go a little bit through some of the background around experimental studies and what we know. So animal studies have really led us to understand how these chemicals impact the immune function. And without going into too many details, this slide really just tells you that PFAS and PFOA are the two compounds, the PFAS congeners that we are most familiar with, that we've studied the most. And this is where we have the most amount of experimental data on from rodents and animals. There's really very limited evidence for other compounds, unfortunately. They just haven't been studied. And we do know through these animal studies that some of the long chain PFAS, as I mentioned, can really alter the immune status in these animals. Things that we've observed in animal studies include things like decreased spleen and thymus weights, and those are organs that are involved in producing cells for the immune system, a reduction in the circulating immune cells in these animals, and a reduction in the antibody levels have also been observed, as well as things like cytokine production, which mediates inflammation in a body. So all important features of your healthy immune function and different parts of your immune function. These have all been associated with, these outcomes have all been associated with increased exposure to these animals, to these long chain PFAS that we've studied. So we'll go to the next slide. Now, what do we know about about epidemiological evidence? So epidemiological evidence is human evidence, evidence in adults or children, pregnant women, et cetera, et cetera. And we study groups of people to be able to understand, based on the animal studies, how we can see the effects in humans. And what we've observed is that antibody levels, both prenatally or in childhood, as a result of PFAS exposure, is negatively associated with specific antibody levels in childhood. And this is our most robust evidence. And this evidence has really been produced by Dr. Grandjean's evidence in the Faroese Islands, produced by Steep and others. And this evidence has been used to help guide the European bodies, regulating bodies, around safe limits to these chemicals. But we've also found that there are other laboratory markers of immune function that have been affected in humans. And this includes things like proteins that respond to infection and inflammation, an increased level of certain types of cells among children that are important for immune regulation. What else have we found is that there's also a higher risk of things like common colds, infections, upper respiratory airway infections in children. And this body has been growing, this body of evidence has been growing, and it's still really in its infancy, and we need to better understand it, better study it, and understand how these chemicals and which one of these chemicals really have the greatest impact on immune function in children. So next slide, I'm going to present just briefly, and the next slide again, a new study that is under review currently, where I used with my team the National Health and Nutrition Survey, the NHANES study, which is a national survey across the United States. And it measures health and nutritional status every 2 years. And we use they use questionnaire interviews, physical examinations, and they collect blood and urine samples from the participants. And in this particular study, we included children between the ages of 3 and 11 and adolescents between the ages of 12 and 19. And this was data that we collected or accessed from cycles 2013 and 2014. So this is a national representative sample across the United States on chemicals. We could go to the next slide. On the exposure, which is, we looked at the serum concentrations of the following PFAS compounds, so PFOA, PFAS, PFHXS, and PFNA. So 4 particular congeners of PFAS. And we looked at the outcome, which was common cold obtained by parent report of their child or their adolescent. And the question that the parents had to answer was, did your child have a head cold or chest cold that started during the previous 30 days? Okay, so the exposure were these 4 PFAS compounds that were measured in the blood of these children, adolescents, and the outcome was common cold or head cold or chest cold in these children, adolescents. And we can go to the next slide. So what did we find? We had about how many children did we have? 517 children between 3 and 11 and 394 adolescents between 12 and 19 years of age. There was a mix between non-Hispanic whites, which was the majority, about 50 to 55%, and the non-Hispanic Blacks were about 15% in the population. And the prevalence of common cold ranged between 17% and 23% in adolescents and children, respectively. So we can go to the next slide. So what did we find in this study? So interestingly enough, we found that after adjusting for factors like age, sex, race, and income level or income to poverty ratio, we found that there was an increased risk of common cold, especially among children, for 3 congeners. So the PFOA, the PFHXS, and PFNA. There was an elevated risk of about 30% on average. So for every doubling of exposure, a child's risk of common cold in the last month increased by about 30%. And this was really most informative for the children between 3 and 11 years of age. We didn't find so much of an association for the adolescents. So this tells us, we can switch to the next slide, and then one more. Yep. So what did we find in the study is sort of conclusions to the study or results of the study, that we found a robust association for serum concentrations of PFHXS and an increased odds of common cold among children and somewhat around adolescents, but less so. And then we also found potentially a positive association between serum PFNA concentrations and common colds in children with, when we did a stratified analysis by gender, we found that the boys seem to have a higher risk than girls. So there might be some sex-specific effects that we have to examine a little bit more deeply. So we can go to the last slide, conclusion. So the association between PFAS and common cold is most evident during childhood. Early childhood may be a critical period for PFAS-related immune effects compared to adolescence. And we know that potentially adolescent PFAS exposure might be more prone to confounding by dietary personal care product use and other exposure sources. And so we didn't really find a strong association among adolescents, but it also might be due to other confounding factors. And that might be the reason why we didn't pick up a clear trend in that group. So we can go to the last slide. So that concludes my my presentation as far as the evidence. And I would like to just spend just a couple seconds acknowledging the funding that I've received for my work, the STEEP program, URI, the Silent Spring Institute, colleagues, Dr. Lochman and Grandjean and other STEEP members. And well, the last slide is my thank you slide. So thank you very much for inviting me here today to present these results. And if you would like to contact me, you can contact me by email, but I'm happy to take questions after Philippe's, Dr. Grandjean's presentation. Thank you very much.
Okay, Dr. Grandjean.
Hello, it's nice to meet you. I'm happy to be here with my good colleague, Dr. Masserlian, and with Cheryl, Mark, and Sarah, who organizes all the slides and all. I wanted to show you this background here because it's from the Faroe Islands, and I like birds very much, and those that you see on the left-hand side are very well known in the northern part of New England, but they also occur in the Faroe Islands, which is just across the North Atlantic, north of Scotland. Now, the reason I am concerned about the perfluorinated compounds are very similar to those that have been summarized by Dr. Masserlian. And what we were particularly concerned about was that some animal experiments that have been carried out about 2008 showed that PFOA in particular could result in a weakened antibody response in mice. And so we thought that we better look and see if this is possible to occur in humans. And just to illustrate to you how the immune system works in this regard, here you have a very simplified map of the various cells involved in the immune system responses. On the very top here, you see the antigen, which is often a protein, and proteins also used in the vaccines. So they trigger a response both from the T cells and from the B cells. And the B cells are the ones that turn into plasma cells and generate antibodies. And those antibodies are specific for the particular pathogen, the particular protein that is circulating in the blood. And therefore, it's crucial for us that the vaccine, if we're using a vaccine, can trigger exactly this response here and generate the right antibodies. So we wanted to try and see if the PFASs had an effect in adults, and we recruited 12 colleagues from a major hospital, and they agreed to be vaccinated with proteins, namely the routine vaccinations that we give to children that just about everybody gets against tetanus and diphtheria. And you can see this is a slide that shows you the number of days. We follow them with blood samples every few days and measured the concentration of the specific antibodies against, this slide shows tetanus. And you can see it doesn't take very long, about a week or so, and then the antibody concentration shoots up. But there's a problem here because one of my colleagues didn't really respond to the vaccine. And it wasn't that we didn't hit the shoulder the right way. We certainly did. And it's just that that person didn't really have a good response. And when we looked at the steepness of the curve, we found that the more PFAS that they had in the blood, the flatter the curve. This was a real eye opener. And so we decided to go much further, and we looked at children to see if this could be replicated. And this is then diphtheria. And both of these scales here are logarithmic. And to summarize the results in brief terms, for every doubling of the child's exposure to PFAS, that is a concentration in the blood, the child was losing about 50% of the specific antibody concentration. And we measured this at age 7. The kids had been inoculated, boosted at age 5. So it's 2 years after the booster, and we saw this very strong association. And we did some additional studies later on and found out that it's particularly the prenatal exposure to the PFAS and the early postnatal exposure that primarily comes from breastfeeding. It was particularly the concentration in the blood at age 3 months. And that fits very well with what Dr. Masserlian summarized about the development of the adaptive immune system that has to respond to all of those pathogens and foreign proteins outside the uterus. So if I may summarize the development, then immune toxicity was actually already known in 1978. It was discovered in a monkey study. It was paid for by a major PFAS producer. And later on, when a medical doctor did his PhD in the occupational health service at a production plant, he found white blood cell changes in the workers associated with the degree of exposure to PFOA. And then, as I said, then there was a mouse study, and we began to find immunotoxicity in children already in 2012. And the good news is that this has been recognized last fall, by the EU, and immunotoxicity is now considered the critical effect of PFAS exposure. Let me now emphasize that the 2 gray studies here in the very top were not published. I would have loved to know this in 1978, but I didn't find out until about 30 years later in connection with this mouse study. We didn't know. And therefore, we are late in realizing what these compounds do to the immune system. And in the meantime, these PFAS compounds have been spread globally. And we all can, I've never in my lab, I've never met a blood sample that did not contain PFAS. We are all exposed. So let me turn briefly to the COVID-19 that worries us all to a great degree. And what we've seen in multiple studies is that the people who get particularly sick are oftentimes the elderly, and men are more likely to get a more severe disease than women. And also there's a risk factor in the kind of pre-existing disease that you have, like diabetes or obesity, is also an important factor. And the interesting thing is that we've seen in many studies that the people who have the highest concentrations of PFAS in their blood are actually the elderly and men, and also postmenopausal women. We also know that diabetes and obesity are associated with elevated exposures to PFAS. So this kind of jives with the risk factors for COVID-19, and we therefore decided to do a study under the difficult circumstances of the close down and being locked out of the university. But we managed to do a study in Denmark where we obtained blood samples from the hospital laboratory, the excess volume from diagnostic tests. And we could do that with all kinds of approvals without knowing the identity of the patients. And actually, more than 100 of these subjects never went to the hospital. So we had a scale, 17 died. We had a scale of severities. And when we got the anonymized data on these subjects, on their pre-existing disease, their age and sex, etc., we could see that we could try to isolate the effects of the various PFAS compounds that we could analyze in the plasma samples. And here's what we found. It was actually very surprising at the background exposures in Denmark that there's no production of PFAS, and we've never seen such high levels in drinking water or in blood samples as have been seen here on Cape Cod. So this is a low background. And what we saw was the only one that showed an association with severity of the disease was the PFBA. The B stands for butane, so it has 4 carbons, as you can see here. It's a short chain PFAS, and it's often considered a precursor, or nowadays I think it's also being used as a substitute for some of the legacy PFAS compounds that oftentimes have 6 or 8 carbons. So we saw a highly significant association there, and we asked ourselves, how could that be? And then we went to an autopsy study that was done in Spain and looked at what they had found in about 20 patients or 20 subjects who had died, suddenly died. And they analyzed lung tissue, and you can see the various PFAS compounds here. And this one here is PFBA, and it was detected in every single lung sample at much, much higher concentrations than the PFAS compounds we normally see in the blood. So this may be the reason why PFBA suddenly showed up as highly significant in regard to the blood concentration, because that was the only thing we could measure in this study. And it does fit with our knowledge of PFAS compounds being immunotoxic. So here's what we know. Industrial compounds are not necessarily, they're certainly not routinely tested for toxicity to the immune system. We know from studies of both adults and children that there are decreased antibody responses at background exposures. We know from prospective studies where we called the mother, we actually sent her an SMS every 2 weeks and asked her if the child had a fever during the last 14 days and what the symptoms were. There was sort of, you could you could check the symptoms that the child had and indicate the number of days. Anyway, we know very well that PFAS exposure in children is positively related to the number of infections that the child gets. And we've also recently published a paper where we showed that the likelihood of being hospitalized with infectious disease increases the more PFAS the child has been exposed to. And that is particularly significant with pneumonia. And that again fits with what I said before about COVID-19. So we have some limited evidence on an association with the severity of this infection. And our current worry is, of course, what about the vaccination? We don't know. But I would say I am worried. And I think that we need to do studies on immune responses to those different vaccinations that are currently being used to see if the antibody response is appropriate, if it is at a significant and an appropriate magnitude to protect against COVID-19, because my suspicion is that that may not be true if people have been exposed in the past or currently to elevated amounts of PFAS, for example, in the drinking water. So I just wanted to give you a status of where we are. As you can see, EPA had very high exposure limits for drinking water levels of the two major PFAS compounds. They lowered that guidance level to 70. The Commonwealth of Massachusetts has a level of 20 now, which is a sum of major compounds. But that last fall, when the European Food Safety Authority updated their risk assessment, they said that the tolerable weekly intake should be no higher than 8 nanograms per kilogram of body weight, which corresponds to 2.2 ppt in drinking water. And if you compare this to what EPA has, it's a factor of 32. I certainly hope that EPA is going to update their risk assessment in the very near future. So I just wanted to summarize here at the very end that both Dr. Masserlian and I and our colleagues from the various institutions, we work with the STEEP Center, and our vision is, of course, to avert human health impact of these exposures and also to disseminate the lessons that we have learned during so far the 4 years of this center's existence. So I wanted to finish my presentation by thanking you all for being here, and we are ready to answer your questions. Thank you.
Thank you so much, Philippe and Carmen, for those really excellent educational presentations. We are honored. Thank you very much. We're going to now open up for a discussion with our panelists. Both of our speakers will address questions from the audience, and we'll also be joined by Amanda Hernandez, a researcher at Silent Spring Institute, working with STEEP's Community Engagement Corps. Thank you to those that have submitted questions already. And our first question is, Carmen, what is worse, PFAS exposure before birth, during childhood, or later on?
That's a great question. I'm a little tiny bit biased with the response. I think, all exposure is harmful to a certain degree depending on when it occurs. The timing of exposure is critical determinant of disease outcomes and adversity. So all windows of exposure are relevant and important. I do believe that the exposure in pregnancy has the potential to have long-term impact on the health of the offspring. It can affect the pregnancy, the woman herself. It can affect the fetus. It can affect how the child develops and grows and how their immune system and other parts, other organs and other functions develop over time during infancy and childhood and all the way up to adults. So we know that there could be long generational effects, that the effects can happen across the life course. And so if I was to choose a window to modify or to change or to reduce as an intervention, I would choose to reduce exposure in pregnancy. And I, in fact, would start to think about exposure before pregnancy. So once someone's pregnant, there's little we can do to sort of modify exposure. We know that these chemicals are long-acting chemicals. They stay in the body for a very, very long time. And so thinking about exposure well before pregnancy and trying to reduce exposure if in the areas that we do have some control. We don't have tremendous control over how we are exposed. But things like drinking water and protecting our drinking water is something we have some control over. And thinking about those mitigation strategies well before pregnancies is initiated in the planning phases, even the years before pregnancy is planned, would be my strategy. I don't know if Dr. Kangin would agree or disagree, but he could chime in.
Well, I would say it depends on the outcome. It depends on the organ system that you're concerned about. But I would agree the prenatal period is important because that's where the programming occurs. That's where the organ development occurs, where we essentially, some of our later disease risks are laid down. But what we're seeing with the immune system is that it's particularly the early postnatal exposure, because a mother can, during her, let's say, 6 months of breastfeeding, she can transfer about half of her body burden to the child, who is, of course, it's a much smaller body. And so the child can get up to what we have seen is something like 10 times higher blood concentrations than the mother had. And so I'd say, and this is exactly what the European Food Safety Authority is saying, we need to protect the current fertile women against PFAS in order to protect the immune system of the next generation.
Thank you.
Can I just add something, Cheryl?
Mm-hmm. Please.
Thank you. Dr. Masserlian mentioned filtering drinking water, and I've seen a couple quick questions in the chat about filtration and whether boiling water removes PFAS. It does not. And I won't go into too much detail about that. I encourage people to check out our last webinar. It's available on the STEEP website, and we'll share the link at the end as well. But in that webinar, we covered drinking water standards, how those standards are set, which PFAS are included in those, and provided some resources on treatment with kind of a regional focus on what's been done on the Cape. So I would encourage you to check that out. And that's kind of for individuals looking to filter their water, regardless of whether you're on public water or a private well.
Thank you, Amanda Hernandez. Another question here for Philippe. Are some PFAS worse than others? And if so, how does the drinking water PFAS on Cape Cod compare?
I'm not an expert on the drinking water on the Cape, but I did check recently if PFBA occurs in the drinking water in the Cape, and it does, but in much smaller concentrations apparently than the other PFAS compounds. And interestingly, it's more in the outer Cape. So it's away from, it's not in Barnstable, it's further out. And so apparently it comes from different sources. And there are apparently individual sources also away from those that you're familiar with from the Joint Forces Air Base and from the fire training area. So if I may say so, I would side with the EU limit, which is 2.2 parts per trillion. It sounds like it's very low, but they wrote the risk assessment, which is incredibly detailed. And I would say anybody who sides with the EPA and the limit or guidance level of 70 must not have recognized the science that has been developed in the most recent years. I hope that answers your question, at least in part.
Thank you, Philippe. Amanda, do you have anything to add to that?
No, I mean, the question was if some PFASs are worse than others and how, I guess, Cape water compares. I'll just say that we've had, with increasing sensitivity and effectiveness of PFAS analysis in kind of both biological and environmental measurements, we can now detect PFASs that we weren't before, but I think Philippe's sense that that lower guidelines, like the 2.2 that he mentioned, are good places for us to aim for. But I'm not sure that the sensitivity methods are there quite yet for the EPA approved method. And so I think that that's a direction that we need to be going in, as well as helping to bridge that gap and help us identify where those PFASs are.
If I may say so, the most of the knowledge we have is on PFOS and PFOA, and that does not mean that they are the most toxic ones. And the studies that have been done looking at other PFAS compounds indicate that others may just be just as bad. And now we have this new study on COVID-19 and the PFBA that I originally thought was close to innocuous, at least not as toxic as the others because it disappears from the blood. But then we saw, hey, it concentrates in the lungs instead. And so I think we still have a lot to learn about these compounds. I would side with the EU authority and with the Commonwealth of Massachusetts that say, let's put them all together for simplicity. We don't know better. We can't have individual limits for all sorts of compounds. We don't know enough. So let's have a joint and very strict limit. I like that. That is very much in agreement with science, I believe.
Thank you. Comment, are there any studies that correlate mid-Cape Yarmouth water quality? Children of mothers who have breast cancer, immune deficient children, or a combination. Are there health studies ongoing on Cape Cod?
Thank you for that question. I saw that in the Q&A box, and it was concerning to me when I read it. We don't actually have any particular studies or focusing on that particular question or that particular group of people that are at risk. So this is a question about mothers that have breast cancer and potentially their children also having some sort of immune compromised state. I think, that's very exploratory in the sense that anecdotally, it could make sense that there could be an association between those two outcomes, breast cancer in the mother and then the child being born immune compromised. They might have a shared exposure time frame. Maybe exposure during pregnancy might predispose a woman both to the outcome of breast cancer as well as her child being at risk of being immune compromised. We just don't have enough data or enough studies to even look at this. Even the number of cases would be small, and so it would be hard to look at. We'd need to look at in a group of people, a larger sample size than what we would find just on that part of the Cape. We would need to accumulate data from a number of parts of the United States to be able to look at that. And it is a question that I'll consider in terms of looking at, things like NHANES data could allow us to look sort of exploratory. It's a cross-sectional study, but nevertheless, it might allow us to, test out hypotheses like this. And I'll bring it back to my team and see what we have in terms of the data, because we do have access to women's information about breast cancer and child health outcomes, so immune outcomes. So thank you very much for bringing that to our attention.
As far as additional studies on the Cape, I think was part of the second question. I'll just share quickly. I think you should be able to see some quick slides about local health studies that are going on the Cape. Philippe, feel free to chime in here if you have additional thoughts about this, but the first is called PFAS Reach, enrolling 120 preschool age children in Hyannis and studying the PFAS effects on immune function in young children. And there's more information here about how you can learn more about that study. And these slides and the recording will be made available, so you can come back to this to reference. And then the second one is called Massachusetts PFAS in Your Health Study, also led by Silent Spring Institute, studying the effects of PFAS in adults and children. This study is enrolling 1,000 adults and 300 children in Hyannis and Ayer, Massachusetts, as part of the CDC ATSDR PFAS multi-site health study. And again, there's more information here on how you can find out more about the study.
Thank you, Amanda. Philippe, is PFBA used in medical equipment that would be used in the intubation of these COVID patients that could contribute to the higher levels detected?
Right, that's a possibility. And what we did was first to analyze all of the blood samples we could get. And then in order to make sure that we had the blood concentration at the time of diagnosis, before hospitalization, we left out all of the blood samples that were taken later on in the clinical development or earlier. And actually, some of the blood samples were taken more than a year earlier, because we wanted to get as many as possible. And most of the PFAS compounds are stable. But when we ignored all of those samples and focused exactly at the time when the subject had been tested positive for corona, then the risk increased up to about 5, so that those with elevated PFBA concentrations had a 5-fold increased risk of getting a more serious course of the infection. So it's possible that there is a PFBA contamination, that it occurs in medical utensils because as part of Teflon or some other material, but this was not what was going on in our study.
Thank you very much. I'm going to ask Amanda and Carmen to please share the answer to this question. This is from a person who lives on Cape Cod, and she knows she's at risk. She has a toddler and would like to have another. And she wants to know what the best filter that she could purchase for their water, and what other actions can she take to limit exposure to PFAS. Carmen, could you go first, please?
I'm going to leave that question maybe to Amanda, because I think the STEEP community engagement group has done some work around this, recommending certain types of water filtration systems that I think are recommended. In terms of other sources of exposure besides water, I think what we focus on, the things that we have control over, we can't control necessarily, what's in our fish or meat. Unfortunately, that's just, the nature of the accumulation of these chemicals up the food chain. But things like packaged food, pizza boxes, popcorn containers in the microwave, Teflon pans, any type of paper that's grease-proof or waterproof that you would get takeout food in, or hamburgers in, or anything that's processed and packaged in paper, most likely has one or more compounds of these PFAS compounds in the paper to make it waterproof, stain resistant, oil proof. All those kinds of features are why we use these chemicals. They allow those products, paper products, to become impermeable. For example, pizza boxes, papers that you have your burgers in at McDonald's, and not just McDonald's, all kinds of brands, even things that you buy at your home. So eliminating sort of processed foods, things that are highly processed in your diet, I think is something that we have some control over. And that's what I would focus on if I was in, that's what I do focus on as a woman, as a mother. And I would encourage people to, stick to the most healthiest nutrition that you can afford. It's obviously more expensive to have better nutrition, but making healthy choices on reducing processed foods, increasing fruit and vegetable consumption, having more of a vegetable-based diet, I think is a healthy way to live for many reasons. And it can also potentially reduce your exposure to these chemicals. Philippe might have more to say as well. He's the true expert on the table here. I'm learning from example.
Well, let me just add that some of the European countries have started banning PFAS compounds, like in pizza boxes, in food wrapping materials. That's what they call it. And so it is possible to have safe food packaging without PFAS. And I think if that's possible, I think that's the way to go. And so I think that we should together demand that PFAS are used as little as possible if they are close to what we consider our daily food or our drinking water. There may be lots of possibilities for using these compounds in closed systems or for purposes where they do not create environmental contamination or human exposure. And that's not what I'm talking about, but I'm talking about our approaches to limiting exposures. I wanted to add one tiny aspect, and that is we don't know yet how to eliminate these compounds from the body. But we do know that they occur in substantial concentrations in the blood. And so women have this advantage that men don't have, that they, and in other terms, you might call it a disadvantage. But in regard to the PFAS, women in fertile age groups will lose a little bit of blood every month. And with that blood, some PFAS leaves the body. And that is probably the main reason that women have lower concentrations of PFAS in the blood than men do. I'm not recommending that you all become blood donors, but in principle, being a blood donor and donating a pint of blood on some occasions will actually decrease your body burden of PFAS. Whether the Red Cross can use that blood is a different story if you have high exposure.
Thank you. Amanda, would you like to share your thoughts about water filtration, water filters?
So I'll just say that drinking water wells on the Cape with specific known contamination and the public water supply have been addressed and taken offline, and treatment is put in place to meet the Massachusetts drinking water guideline. You may still want to take on additional water treatment, or you have a private well. As far as filtration, reverse osmosis and activated carbon have both been shown to be effective at removing PFASs. But there are specific independent certifications to look for. And those resources, I believe we're going into significantly more detail on our previous webinar. So I'm not trying to differ, and just think that that's a really great resource for the community as well. And our STEEP website also has several handouts specifically on water treatment, specifically for private well owners. So you can find those on our website as well, and we'll share the URL again at the end.
Thank you very much, Amanda Hernandez, Dr. Carmen Masserlian, Dr. Philippe Grosjean, for these excellent presentations. Again, honored and so grateful to you for being here with us today. On behalf of the STEEP team, I'd like to thank you again for joining us. Thank you to our speakers for all the great information and conversation. Many of you have heard me say this before over and over again. We're doing this work to make the world a better place for our children, our grandchildren, and for future generations. We're deeply appreciative to the town of Barnstable for their support of this program, and to the STEEP Research Translation Corps and Community Engagement Corps for continuing to share their knowledge and engage the community in this critically important issue. A recording of this webinar will be made available on the town of Barnstable's website, as well as the STEEP website. Unfortunately, we're not able to get to all of your questions today, but our team has captured them all, and we will do our best to answer them in future webinars. As with this webinar, future webinars will support not only Cape Cod, but also affected communities nationwide by sharing information and offering solutions so that communities can better protect themselves. We invite you to please join us for our next webinar on April 14th, which will focus on essential uses of PFAS. Thank you, everyone. Have a great day.